Hi, morning everybody. Today our topic of discussion would be the mechanism of oxidative phosphorylation. So, most of the usable energy obtained from the breakdown of carbohydrates or fats is derived by oxidative phosphorylation, which takes place within mitochondria. For example, the breakdown of glucose by glycolysis and the citric acid cycle yields a total of 4 molecules of ATP, 10 molecules of NADH, and 2 molecules of FADH2. Electrons from NADH and FADH2 are then transferred by molecular oxygen coupled to the formation of an additional 32 to 34 ATP molecules by oxidative phosphorylation. Electron transport and oxidative phosphorylation are critical activities for protein complexes in the inner mitochondrial membrane, which ultimately serve as the major source of cellular energy. Right? Now we go to the next topic, will be electron transport. During oxidative phosphorylation, electrons are derived from NADH2 and FADH2. Know these points, right? And combined oxygen and the energy released from these oxidation reduction reactions is used to drive the synthesis of ATP from ADP. The transfer of electrons from NADH to oxygen is a very energy yielding reaction with the delta G0, that's the Gibbs free energy, at or being equal to minus 52.5 kilocalories per mole for each pair of electrons transferred, right? Delta G0, okay, that's the notation, Gibbs free energy. Here we're using the capital delta, right? The uppercase. To be harvested in usable form, this energy must be produced gradually by the passage of electrons through a series of carriers which constitute the electron transport chain. Right? I hope you know what the electron transport chain is. It's exactly what we're speaking of now. Right? Just to give you a small depiction of what exactly electron transport chain is, just want to give you a small note on that. Explanations on that particular thing, these keywords will be given at the end of the chapter, right? Now, these carriers are organized into 4 complexes in the inner mitochondrial membrane. A 5th protein complex then serves to couple the energy yielding reactions of electron transport to ATP synthesis. Now, electrons from the NADH enter the electron transport chain in complex 1. I told you there are 4 complexes. In complex 1, which consists of nearly 40 peptide chains. These electrons are initially transferred to NADH to flavin mononucleotide and then through an iron sulfur carrier to coenzyme Q, an energy yielding process with the delta G0 equal to minus 16.6 kilocalories per molecule. Coenzyme Q, also called as ubiquinone, ubiquinone, is a small lipid soluble molecule that carries electrons from complex 1 through the membrane to complex 3, which consists of about 10 polypeptides. In complex 3, electrons are transferred from cytochrome B to cytochrome C, an energy yielding reaction with delta G0 equal to minus 10.1 kilocalories per mole. Cytochrome C, a peripheral membrane protein bound to the outer surface of the inner membrane, then carries electrons to complex 4, which is through cytochrome oxidase, where they finally transfer to O2 with the depiction of delta G equal to minus 25.8 kilocalories per mole. Now, transport of electrons from NADH, all the diagrammatic expressions, illustrations will be shown to you after I finish the lecture, okay? Or on the next lecture, accordingly as per the time constraints that are there. If there are no time constraints, I'll do it today itself. If not, the next lecture, or by afternoon, you'll have the next video coming up. A distinct protein complex 2, which consists of 4 polypeptides, receives electrons from the citric acid cycle. Intermediate, succinate. These electrons are transferred to FADH2 rather than to NADH and then to coenzyme Q. From coenzyme Q, electrons are transferred to complex 3 and then to complex 4, as already explained to you. In contrast to the transfer of electrons from NADH to coenzyme Q at complex 1, the transfer of electrons from FADH2 to coenzyme Q is not associated with a significant decrease in free energy and therefore is not coupled to ATP synthesis. Consequently, the passage of electrons derived from FADH2 through the electron transport chain yields free energy only at complexes 3 and 4. The transport of electrons from FADH2. Electrons from succinate enter the electron transport chain via FADH2 in complex 2. They are then transferred to coenzyme Q and carried through the rest of the electron transport chain, okay, which I should show you as a diagrammatic representation after this lecture, right? The free energy derived from the passage of electrons through complexes 1, 3, and 4 is harvested by being coupled to the synthesis of ATP. Importantly, the mechanism by which the energy derived from these electrons transport reactions is coupled to ATP synthesis is fundamentally different from the synthesis of ATP during glycolysis and the citric acid cycle. In the latter case or cases, a high energy phosphate is transferred directly to ADP from the other substrate of an energy-yielding reaction. For example, in the final reaction of glycolysis, the high energy phosphate of phosphoenol pyruvate is transferred to ADP, yielding pyruvate plus ATP. Such direct transfer of high energy phosphate groups does not occur during electron transport. Instead, the energy derived from electron transport is coupled to the generation of a proton gradient across the inner mitochondrial membrane. The potential energy stored in this gradient is then harvested by a fifth protein complex, which couples the energetically favorable flow of protons back across the membrane to the synthesis of ATP. Now we come to the next topic called chemiosmotic coupling. The mechanism of coupling electron transport to ATP generation, chemiosmotic coupling, is a striking example of the relationship between structure and function in cell biology. Right? The hypothesis of chemiosmotic coupling was first proposed in 1961 by Peter Mitchell, who suggested the proposed hypothesis, which later was accepted as a theory. He had suggested that ATP is generated by the use of energy stored in the form of protein gradients across biological membranes, rather than by direct chemical transfer of higher energy groups. Biochemists were initially highly skeptical of this proposal. Initially, it was spot on disputed, but later on, another scientist by the similar name, Peter, about 55 years later, had proved him right. And that's when Peter Mitchell's hypothesis became accepted as a theory. Right? In most cases, it's like that. You first have your first and second line of defense, and then you go for a final defense because it's your thesis. You have to prove it to them, right? Or to the examining board. And now the overwhelming evidence eventually accumulated in its favor, and chemiosmotic coupling is now recognized as a general mechanism of ATP generation, operating not only in mitochondria but also in chloroplasts and in bacteria, where ATP is generated via a proton gradient across the plasma membrane. Electron transport through complexes 1, 3, and 4 is coupled to the transport of protons out of the interior of the mitochondria. I think I've shown all this in the diagrammatic representations in my last lecture, where we dealt with what photosynthesis and the activities, there's various cycles in it, all right? Well, I'm showing you this diagram again, a diagrammatic representation, so you get an idea of what exactly it is. Consequent to each class or on the next day of lecture, I'll give you sessions on question and answer sessions, and we'll have diagrammatic illustrative sessions. I have done a couple of them here, maybe 2 or 3 on one is on citric acid cycle, the other on Calvin cycle, and another one photosynthesis, right? So I'll give you diagrammatic representations after this theoretical explanation, right? Okay. Now, the energy yielding reactions of electron transport are coupled to the transfer of protons from the matrix to the intermembrane space, which establishes a proton gradient across the inner membrane. Complexes 1 and 4 appear to act as proton pumps that transfer protons across the membrane as a result of conformational changes induced by electron transport. In complex 3, protons are carried across the membrane by coenzyme Q, which accepts proton from the matrix at complexes 1 or 2 and releases them into the intermembrane space at complex 3. Complex 1 and 3 each transfer 4 protons across the membrane per pair of electrons. In complex 4, 2 protons per pair of electrons are pumped across the membrane, and another 2 protons per pair of electrons are combined with oxygen to form water, that's H2O, within the matrix. Thus, the equivalent of 4 protons per pair of electrons are transported out of the mitochondrial matrix at each of these 3 complexes. This transfer of protons from the matrix to the inner membrane space plays the critical role of converting the energy derived from the oxidation-reduction reactions of electron transport to the potential energy stored in a proton gradient. Because protons are electrically charged particles, the potential energy stored in the proton gradient is electric as well as chemical in nature. The electric component corresponds to the voltage difference across the inner mitochondrial membrane within the matrix of the mitochondrion negative and the intermembrane space positive. The corresponding free energy is given by the equation delta G is equal to minus F delta V, right? Where F is the Faraday constant and delta V is the membrane potential. Delta is uppercase. Don't forget that. The additional free energy corresponding to difference in proton concentration across the membrane is given by the equation. Just write it down while I tell you. Uppercase delta G is also in capitals is equal to RT. RT would be actually 8.314 joules per kilogram mole, right? Joules per k mole. So RT ln H plus I by H plus 0. We'll be showing all this, all right? It's note it down so you get to know what it is. Where H plus I and H plus 0 refer respectively to the proton concentrations inside and outside the mitochondria. In metabolically active cells, protons are typically pumped out of the matrix such that the proton gradient across the inner membrane corresponds to about 1 pH unit or a tenfold low concentration protons within mitochondria. The pH of the mitochondrial matrix is therefore about 8 compared to the neutral pH approximately 7 of the cytosol and intermembrane space. This gradient also generates an electric potential of approximately 0.14 volts across the membrane with the matrix negative. Both the pH gradient and the electric potential drive protons back into the matrix from the cytosol. So they combine to form an electrochemical gradient across the inner mitochondrial membrane corresponding to uppercase delta, capital G, that's delta G, of approximately minus 5 kilocals per mole per proton, right? Because the phospholipid bilayer is impermeable to ions, protons are able to cross the membrane only through a protein channel. This restriction allows the energy in the electrochemical gradient to be harnessed and converted to ATP as a result of the action of the fifth complex involved in oxidative phosphorylation, complex 5 or ADP synthase. ADP synthase is organized into two structurally distinct components, F0 and F1, which are linked by a slender stalk. The F0 portion spans the inner membrane and provides a channel through which protons are able to flow back from the intermembrane space to the matrix. The energetically favorable return of protons to the matrix is coupled to ADP synthesis by the F1 subunit, which catalyzes the synthesis of ATP from ADP and phosphate ions Pi. Detailed structural studies have established the mechanism of ATP synthase action, which involves mechanical coupling between the F0 and F1 subunits. In particular, the flow of protons through F0 drives the rotation F1, which acts as a rotary motor to drive ATP synthesis. They even refer to that as a turbine producing electricity. It appears that the flow of 4 protons back across the membrane through F0 is required to drive the synthesis of one molecule of ATP by F1, consistent with the proton transfers at complexes 1, 3, and 4, each contributing sufficient free energy to the proton gradient to drive the synthesis of one ATP molecule. The oxidation of one molecule of NADH thus leads to the synthesis of 3 molecules of ATP, whereas the oxidation of FADH2, which enters the electron transport chain at complex 2, yields only 2 ATP molecules. The next will be transport of metabolites across the inner membrane. In addition to driving the synthesis of ATP, the potential energy stored and the electrochemical gradient drives the transport of small molecules into and out of mitochondria. For example, the ATP synthesized within mitochondria has to be exported to the cytosol, while ADP and Pi needed to be imported from the cytosol for ATP synthesis to continue. The electrochemical gradient generated by proton pumping provides energy required for the transport of these molecules and other metabolites that need to be concentrated within mitochondria. There are figures for each of these as I talk to you, that will be shown to you. The transport of ATP and ADP across the inner membrane is mediated by an integral membrane protein, the adenine nucleotide translocator, which transports one molecule of ADP into the mitochondrion in exchange for one molecule of ATP transferred from the mitochondrion to the cytosol. Because ATP carries more negative charge than ADP, -4 to -3, this exchange is driven by the voltage component of the electrochemical gradient. Since the proton gradient establishes a positive charge on the cytosolic side of the membrane, the export of ATP in exchange for ADP is energetically favorable. The synthesis of ATP within the mitochondrion requires phosphate ions, Pi, as well as ADP, so Pi must also be imported from the cytosol. This is mediated by another membrane transport protein, which imports phosphate H2PO4- and exports hydroxyl ions OH-. This exchange is electrically neutral because both phosphate and hydroxyl group ions have a charge of -1, that was hydroxyl ions of a charge of -1. However, the exchange is driven by the proton concentration gradient. The higher pH within mitochondria corresponds to a higher concentration of hydroxyl ions, favoring their translocation to the cytosolic side of the membrane. Energy from the electron chemical gradient is similarly used to drive the transport of other metabolites into mitochondria. For example, the import of pyruvate from the cytosol, which is produced by glycolysis, is mediated by a transporter that exchanges pyruvate for hydroxyl ions. Other intermediates of the citric acid cycle are able to shuttle between mitochondria and the cytosol by similar exchange mechanisms. So that's it about the electron transport chain. I'll be giving you a separate lecture on the chemoosmotic theory next class, along with diagrammatic representations, right? So take notes, understand what has been spoken to, make notes and study. If any doubts, connect with me on my email, right? Have a great time. Good day.